Dear students, welcome to today's online session on chromosomal dynamics, where we will explore the structural and functional organization of chromosomes and the molecular machinery that ensures their accurate segregation. Together, we will examine how mitosis and meiosis safeguard genetic stability and diversity, and how cytogenetic techniques help us understand genetic disorders and their consequences. So today, our learning objectives of the topic are to explain the structural and functional organization of chromosomes, including centromeres, telomeres, chromatids, and chromatin states; to analyze the molecular mechanisms of chromosomal dynamics, emphasizing the roles of cohesins, condensins, kinetochores, and spindle fibers in chromosome condensation, alignment, and segregation; to evaluate the process of meiosis and its role in generating genetic diversity through recombination and independent assortment; to demonstrate the proficiency in cytogenetic techniques such as karyotyping, fluorescence in situ hybridization or the FISH, microscopy, and molecular cytogenetics, and apply them in explaining the diagnosis of genetic disorders; to assess the biological consequences of errors in chromosomal dynamics, including missegregation, non-disjunction, and their association with cancer and genetic diseases. First of all, chromosomal dynamics and the cytogenetic basis of mitosis and meiosis explore how chromosomes structurally organize, condense, and segregate to ensure accurate transmission of genetic material during cell division. The cytogenetic basis of cell division lies in the precise movements and rearrangements of chromosomes during mitosis and meiosis. These processes guarantee faithful transmission of genetic information from somatic growth and sexual reproduction. These processes reveal the molecular choreography of cohesins, condensins, kinetochores, and spindle fibers that safeguard genomic stability and underlie growth, development, and reproduction. Chromosomes as a carrier of heredity. Chromosomes are the fundamental units of heredity. They are composed of DNA tightly associated with proteins forming chromatin. Each chromosome consists of a centromere. This region anchors kinetochore proteins linking chromosomes to spindle microtubules during mitosis and meiosis for accurate segregation. Telomeres, protective caps at chromosome ends, repetitive DNA, TTAGGG sequence caps, chromosome ends preventing degradation. Origin of replication. DNA sites where replication starts, ensuring the efficient genome duplication during S phase. Chromatids, identical DNA copies after replication joined at the centromere. The molecular organization of a chromosome. Chromosomal dynamics and the process of mitosis and meiosis is fundamental to grasping how cells divide and pass on the genetic information. These processes govern the cell growth, tissue repair, and reproduction, ensuring genetic stability and diversity. DNA packaging. Eukaryotic DNA, a lengthy molecule, is compactly organized to fit within the nucleus while allowing replication and transcription. The nucleosome, the core unit, wraps around 147 base pairs of DNA around 8 histone proteins, which are the H2A, H2B, H3, H4, compacting in 7 folds. Histone H1 stabilizes this structure. Nucleosomes form a 30 nanometer fiber coiling into the solenoid, further condensing the DNA. Errors in the packaging can lead to diseases like cancer. Euchromatin, a lightly packed and transcriptionally active, euchromatin's open structures allow DNA access for transcription factors and RNA polymerase. Heterochromatin, densely packed and transcriptionally silent, it restricts the DNA access, including centromeres and telomeres. Now we will be discussing about mitosis, cytogenetic basis of somatic division. Mitosis is the process by which a cell divides into two genetically identical daughter cells. It ensures equal distribution of replicated chromosomes. The phases of mitosis. First one is the prophase. Prophase begins mitosis by condensing diffuse chromatin into visible chromosomes, each with two sister chromatids joined at the centromere. Centrosomes migrate and organize the mitotic spindle, a bipolar framework essential for chromosome movement. The nucleolus disappears and the resources are restricted to division. Second is the pro-metaphase. In pro-metaphase, the nuclear envelope breaks down, enabling spindle microtubules to attach to kinetochores at centromeres. These attachments initiate chromosomes congression towards the cell equator. The third is the metaphase. Chromosomes align at the metaphase plate, ensuring balanced segregation. Accurate alignment guarantees genetic fidelity as seen in embryonic development. The fourth is the anaphase. Kinetochore microtubules shorten to pull chromatids to opposite poles, while non-kinetochore microtubules elongate the cell. Motor proteins like kinesins drive this separation. Failures such as non-disjunction cause disorders like Down syndrome. Fifth is the telophase. Telophase restores nuclei as chromosomes decondense into chromatin. Nuclear envelopes reform and spindles disassemble. Sixth is the cytokinesis. Cytokinesis divides the cytoplasm, producing two diploid daughter cells. In animals, an actin-myosin contractile ring forms a cleavage furrow, which in plants, a cell plate develops in a new wall. Regulation of mitosis. Mitosis is tightly regulated to prevent errors. Cyclins and cyclin-dependent kinases, or the CDKs, protein complexes that drive progression through G2M checkpoint, ensures DNA replication is complete. Spindle checkpoints halt metaphase if kinetochores are misattached, preventing unequal chromosome distribution. Tumor suppressors like p53 monitor the DNA damage. Faulty regulation can lead to cancer. Biological significance of mitosis. Growth and development of multicellular organisms. Mitosis enables cell division for growth, producing cells for tissues and organs during development. For example, mitotic division in early embryo expands cell numbers, ensuring proper organ formation. Tissue repair and regeneration. Mitosis replaces damaged or dead cells, facilitating tissue repair. In skin, mitotic division of basal cell heals wounds, while in liver, it regenerates tissue after injury. This process maintains tissue function and integrity. Asexual reproduction in unicellular organisms. In organisms like yeast or amoeba, mitosis enables asexual reproduction, producing genetically identical offsprings. This ensures rapid population growth under favorable conditions. Genetic stability ensures identical genetic content in daughter cells. Genetic instability or the errors in mitosis can cause cancer. Mitotic errors such as non-disjunction or checkpoint failures lead to aneuploidy or chromosomal instability. Uncontrolled mitosis, often due to mutated tumor suppressors like p53, causes cancer by promoting excessive cell proliferation. Chromosomal instability can also result in genetic disorders, for example, Down syndrome. Non-disjunction in gamete formation leads to an extra chromosome 21. Now, we will be discussing about meiosis, cytogenetic basis of sexual reproduction. Meiosis is a specialized cell division in sexually reproducing organism, reducing the chromosome number from diploid, which is 2n, to haploid, which is n, to form gametes, which are the sperm and the eggs. It involves one DNA replication followed by two division, meiosis 1 and meiosis 2, ensuring genetic diversity for reproduction. The key features of meiosis: reduction of chromosome number from diploid to haploid. This ensures the gametes combine during fertilization to restore the diploid number, maintaining species-specific chromosome counts. Genetic recombination through crossing over. In prophase 1, homologous chromosomes exchange DNA segments at chiasmata, creating new allele combination. This recombination, seen in sperm production, enhances genetic diversity critical for adaptation. Independent assortment of chromosomes. During metaphase 1, homologous chromosomes align randomly, leading to varied gametic combination. This shuffling, as in egg formation, contributes to genetic variation. Production of genetic diversity essential for evolution. Crossing over and independent assortment generate unique gametes, driving evolutionary change. For example, diverse gametes enable species to adapt environmental shift, supporting survival. Stages of meiosis. Meiosis comprising meiosis 1, which is the reductional division, and meiosis 2, which is the equational division, reduces the chromosome number from diploid to haploid, producing diverse gametes through two sequential divisions after one DNA replication. First, we will be discussing about meiosis 1, the reductional division, and the first one is the prophase 1, the longest and the most complex phase, subdivided into 5 stages. The liptotein, chromosome condense into thin thread-like structures becoming visible. Next is the zygotine stage. Homologous chromosomes pairs via the synaptonymal complex, initiating synapses. The third is the pachytene. Crossing over occurs where non-sister chromatids exchange DNA at chiasmata, promoting genetic recombination. Next is the diplotein. Homologous, partially separate, revealing chiasmata as crossing over points. Diakinesis. Chromosomes are fully condensed and the nuclear envelope breaks down, preparing for spindle attachment. For example, in sperm formation, crossing over enhances genetic diversity. Next is the metaphase 1. Homologous chromosome pairs align at metaphase plate. Random orientation of each pair, driven by kinetochore microtubule attachments, leads to independent assortment, shuffling maternal and paternal chromosomes. In egg production, this ensures varied gamete combinations critical for genetic variation. Next is the anaphase 1. Homologous chromosomes are pulled to opposite poles by shortening microtubules. reducing chromosome number from diploid to haploid. Unlike mitosis, sister chromatids remain attached, ensuring each cell receives one homologue. This is vital in gametogenesis to maintain proper ploidy. Telophase 1 and cytokinesis. Chromosomes reach poles, often decondensing slightly, and nuclear envelopes may reform. Cytokinesis divides the cytoplasm, forming two haploid cells, each with duplicated chromosomes. In oogenesis, this produces one cell and a polar body, conserving resources for the egg. Next is the meiosis II, or the equational division. It resembles mitosis but occurs in haploid cells where no DNA replication takes place. In prophase II, chromosomes condense, new spindle forms. Metaphase II, chromosomes align at the equator, kinetochores attached to opposite poles. Anaphase II, sister chromatids separate individual chromosomes. Telophase II and cytokinesis. Chromosomes decondense, nuclear envelopes reform, produces 4 haploid gametes, each genetically unique. For example, spermatogenesis yields 4 functional sperm, increases reproductive diversity. Now we will be discussing about biological importance of meiosis. It maintains constant chromosome number across generations. Meiosis reduces chromosome number from diploid to haploid, ensuring gametes such as sperm and eggs have half the chromosomes. During fertilization, haploid gametes combine to restore the diploid number, for example, 46 in humans. This maintains species-specific chromosome counts across generations are seen in human reproduction, preventing doubling with each generation, produces variation through recombination and independent assortment. Crossing over in prophase I and random alignment of homologous chromosomes in metaphase I create diverse gametes, essential for fertility and sexual reproduction. Meiosis produces viable gametes necessary for sexual reproduction. In humans, it ensures functional sperm and eggs supporting fertility. Errors cause aneuploidy, for example, Down syndrome, which is trisomy 21, or the Turner syndrome, which is monosomy of the X chromosome. Non-disjunction during meiosis I or II results in gamete with abnormal chromosome numbers, leading to aneuploidy. Trisomy 21, the Down syndrome, or the monosomy of X chromosome, which is the Turner syndrome, can result causing developmental or health issues, underscoring meiosis precision. Next is the cytogenetic techniques to study mitosis and meiosis. Cytogenetic techniques help visualize and analyze chromosomes during mitosis and meiosis, providing insights into structure, number, and dynamics, as well as aiding in diagnosing genetic disorder. Karyotyping chromosome number and morphology. Cells are arrested in metaphase, stained, for example, GIMSA, and photographed to form a karyogram. This reveals the chromosome number, size, and shape, confirming the human diploid set of 46. Abnormalities like trisomy 21 or the Down syndrome or cancer-related aneuploidy can be detected. Fluorescence in situ hybridization, or the FISH. FISH uses fluorescent DNA probes to bind specific regions visualized under a fluorescence microscope. It identifies translocations, deletions or duplications such as Philadelphia chromosome in leukemia and maps genes during meiosis. Microscopy. Stained tissue like onion root tip in mitosis or grasshopper testes in meiosis allow the visualization of division stages such as metaphase alignment or anaphase separation. Molecular cytogenetics. Advanced methods use fluorescent markers or chromosome painting to track chromosome dynamics, revealing crossing over, segregation errors or non-disjunction linked to infertility and genetic disorders. Chromosome dynamics during the cell cycle. Chromosomal dynamics refer to the structural and positional changes chromosomes undergo during the cell cycle, ensuring accurate replication, segregation, and genetic stability. Interphase. Chromosomes remain uncoiled as diffused chromatin, which permits transcription and DNA replication. In G1, cells grow and synthesize proteins during S phase. DNA replication produces identical sister chromatids, and in G2, cells check for replication errors and prepare for mitosis. This organization ensures the genome is ready for division. M phase includes mitosis and meiosis, where chromosomes condense and segregate through intricate mechanisms. Mitosis. Chromatin condense into compact chromosomes with two sister chromatids joined at centromere. This condensation, beginning in prophase, prevents entanglement and enables smooth alignment at metaphase plate. Kinetochores. Protein complexes at centromeres attach chromosomes to spindle microtubules. Motor proteins like dyneins and kinesins guide separation during anaphase. The spindle assembly checkpoints or the SAC monitors attachments and delays progression until errors are corrected, ensuring equal chromosome distribution. Meiosis. Chromosomal dynamics generate haploid gametes and promote diversity. During prophase 1, homologous chromosomes pair through the synaptonymal complex, stabilizing them for crossing over. At chiasmata, non-sister chromatids exchange DNA, creating recombinant chromosomes and new allele combinations. Cohescent proteins maintain chromatid cohesion until separation, while spindle microtubules ensure accurate segregation. The SAC prevents premature or faulty segregation, guarding against non-disjunction. Errors in recombination or chromosome pairing may cause infertility, genetic disorders, or aneuploid conditions such as Down syndrome. Centromeres and kinetochores are central to both mitosis and meiosis. In mitosis, sister chromatids attach to opposite poles, while in meiosis 1, homologous chromosomes attach to the same pole, ensuring proper reductional division. The mitotic spindle, composed of kinetochore and non-kinetochore microtubule, orchestrates chromosome movements and cell elongation. Regulation of chromosomal dynamics. Chromosomal dynamics are regulated by cohesins, condensins, and motor proteins, microtubules. Chromosomal dynamics during cell divisions are tightly regulated by cohesins, condensins, and motor proteins interacting with microtubules, ensuring precise chromosome organization and segregation. Cohesins holding sister chromatids together. Cohesins are ring-like protein complexes made of SMC proteins and subunits such as SCC1 that hold sister chromatids together after DNA replication. In mitosis, cohesin maintain cohesion until separase cleaves them at anaphase. In meiosis, arm cohesins are removed in meiosis 1 to separate homologs, while centromeric cohesins are cleaved in meiosis 2 for chromatid separation. Cohesin dysfunction can cause non-disjunction and aneuploidy, as in Down syndrome. Condensin chromosome condensation. Condensins, also SMC based, compact chromatin into condensed chromosomes during prophase. By looping DNA, they shorten chromosomes, preventing entanglement. Working with topoisomerase 2, condensins ensure smooth segregation and precise alignment at the metaphase plate. Mutations disrupt condensation, leading to genomic instability and cancer. Motor proteins interacting with microtubules. Kinesins and dyneins regulate chromosome movement along microtubules. Kinesins drive poleward movement at anaphase, while dyneins assist in kinetochore capture. Dynamic microtubules guided by these proteins ensure accurate segregation in mitosis and meiosis. Malfunctions cause aneuploidy or arrest. Now we will be discussing about the applications and relevance of chromosomal dynamics. The study of chromosomal dynamics inform medicine, agriculture, biotechnology and evolution. Diagnosing genetic diseases. Cytogenetic tools like karyotyping and FISH identify chromosomal disorders such as Down syndrome, Klinefelter syndrome, and cancer-specific translocations such as BCR-ABL in leukemia. Prenatal screening detects aneuploidy, guiding interventions. Antimitotic drugs targeting cell division enhance cancer therapy. Plant breeding and hybrid development, understanding recombination and mitotic behavior allows crop improvement. Manipulating crossing over enhances hybrid vigor, as in maize. Cytogenetic screening identifies chromosome variants, supporting resilient, high-yield crops vital for food security. Biotechnology, gene editing, cloning, transgenics. Insights into chromosome behavior enable precise genome manipulation. CRISPR-Cas9 targets chromosomal DNA, while mitotic fidelity ensures stable cloning outcomes. Transgenics, such as insulin-producing bacteria, showcase medical applications of chromosomal engineering. Evolutionary biology as a source of variation. Crossing over and independent assortment generate diversity, driving evolution. These mechanisms create novel allele combinations critical for adaptation, as seen in antibiotic-resistant bacteria. Studying recombination rates illuminates evolutionary trajectories across species. Dear students, as we conclude, remember that understanding chromosomal dynamics not only explains how life maintains stability and diversity, but also connects directly to medicine, biotechnology, and evolution. I encourage you to reflect on these concepts as essential foundations of your future studies and research in biology. Thank you.